Howdy Pima Aztecs, and welcome to Module 2A, Chapter 2, Biochemistry Part 3. And there will be a Biochemistry Part 4, so you have that to look forward to. So, remember God. What is God in biochemistry? Shown: carbon, hydrogen, oxygen, and nitrogen. So those are the major elements that make up living things. So this is just a pattern of chemical reaction review. I did this on the last video, but I want to go over it again because it will be important for this video. So there's different types of chemical reactions, a lot of different types, but important for physiology is a synthesis reaction. So in a synthesis reaction, smaller particles are bonded together to form larger complex molecules. So example is amino acids. And we're going to talk about this amino acids and proteins, not in this video, but the next video. But amino acids are joined together to form protein molecules. So this would be a synthesis reaction where you took every one of these little amino acids and you link them up with a chemical bond and you'd make a giant protein molecule. Decomposition reaction, an example would be when bonds are broken in a large molecule. So this is, we'll talk about this guy in this video, glycogen. Glycogen is the storage form of glucose. So glycogen is a big molecule, so it's a large molecule, and you can break it down when you need glucose. So each one of these little green things is a glucose molecule or a glucose monomer, and they're all linked together, form glycogen. And when you are low in blood sugar, your glycogen gets broken down into glucose, and then glucose can be taken up by your cells and used to make, say it, ATP. Cool. All right, so carbohydrates. Let's talk about the major molecules that make us up and that we like to eat. Carbohydrates contain carbon, hydrogen, and oxygen. So here's a flow chart of the way we can look at carbohydrates. If you've had a nutrition class, you know this. So the major category of macromolecule is a carbohydrate. You can break carbohydrates down into simple carbohydrates and complex carbohydrates. If you are a simple carbohydrate, you can be a monosaccharide. So saccharide, it just means sweet in Greek. So single molecule would be a monosaccharide, and when you have two single monosaccharides linked together, you would be called a disaccharide. And here's some examples of monosaccharides: glucose, fructose, and galactose. Here's some examples of disaccharides: maltose, malted milk balls; lactose, lactose-free drinks; and sucrose. Sucrose is table sugar. Polysaccharides would be under complex carbohydrates. Polysaccharides would include your starches, delicious potatoes, fibers, and glycogen. And we just saw that glycogen molecule. Carbohydrates are organic compounds. It's important to note that 60 to 80% of the volume of living cells consists of organic compounds. And then this guy, C6H12O6, this, I'm so sweet. It's a glucose molecule. So we just talked about this. Carbohydrates can be classified according to their size. A monosaccharide is one simple sugar. Disaccharide is two monosaccharides put together. And a polysaccharide is a long branching chain of simple sugars. And then this is a general term, a polymer. You may have heard of the term polymer in other areas of science, but in anatomy and physiology, a polymer is just a chain-link molecule made of similar or repeating units called monomers. So for example, a carbohydrate is a polymer, and it's made of the monomers glucose and maybe fructose and galactose. Many biological molecules are polymers, so carbohydrates and proteins are classic polymers. And then this just shows the disaccharide maltose is made up of glucose and another glucose. Lactose is made up of glucose and galactose. And sucrose, delicious table sugar, is made of glucose and fructose. So here's a cartoon of a simple sugar or a monosaccharide. So a monosaccharide is the monomer of carbohydrates. Monosaccharides can be, usually it's a ring-like structure like this, contains 3 to 7 carbon atoms. In each corner, it doesn't show this, but if you've had chemistry, you know this. In each corner here, there's a carbon atom. It just doesn't show it. And if it doesn't show it and it puts an O there, that means an oxygen is there. So examples of monosaccharides are glucose, fructose, galactose, ribose, deoxyribose. And then disaccharides, we said, were it's called a double sugar or disaccharide. It's when 2 simple sugars are hooked together by a bond. And the examples we saw in the previous slide: maltose, lactose, sucrose, delicious. Glucose and fructose hooked together is sucrose. So here's a cartoon of a polysaccharide. It's a starch like you'd find in potatoes, for example. It's just a long chain in this cartoon of glucose monomers. So here's a glucose monomer. Here's a glucose monomer. Glucose monomer. Glucose monomer. Glucose monomer. When they're all joined together in this long chain, it's called a starch, or you could call it, generally it's a polymer, because it's a whole bunch of monomers hooked together. And then just here's the chemical structures of the monomers that could be hooked together to form polymers. So you could have in sugars, you could have glucose or fructose. Here's what galactose looks like chemically. So polysaccharides include starch and glycogen. Polysaccharides are large. They're insoluble, so they don't dissolve in water. And they function as storage products. So this is, for example, starch is a delicious storage form of glucose. We talked about these reactions in the previous video, but let's look at it again. Look at them again. This is a dehydration synthesis going from left to right, and hydrolysis reaction is going from right to left. So if you're doing a dehydration synthesis, you're taking these 2 monomers, glucose and fructose. So it kind of looks like they're linked here, but they're not. That's just showing you these 2 hydroxyl groups in blue. So if you're doing a dehydration synthesis, you want to make sucrose, delicious table sugar. You pull a water molecule out of these 2 monomers. You hook them together and make sucrose, and then you also get a water molecule out of that. If you want to take sucrose and break it down into glucose and fructose, so that means you're going right to left, then you have to put in a water molecule to break this bond, and then you'll have the 2 monomers. Okay, that was it for carbohydrates. How about lipids? As you know, fat, or lipids, is a great source of storing energy. Examples of lipids would include triglycerides, fatty acids, cholesterol, delicious cholesterol. The ratio of fatty acids to glycerol in a triglyceride, this cartoon down here, is 3 to 1. So there's 3 fatty acid chains. So here's 1 fatty acid chain. Here's another fatty acid chain. Here's another fatty acid chain. And then here's the glycerol head group or molecule. And you hook those together and you make a triglyceride or a neutral fat. And then one of the products is water, 3 water molecules. Lipids contain CHO, carbon, hydrogen, and oxygen. Lipids are insoluble in water, but they're soluble in other lipids, so lipids can dissolve in other lipids. And you can, well, a lipid is composed of 3 fatty acids, like we just looked at in the cartoon, and 1 glycerol molecule. And if you are a fatty acid, you could be a saturated fatty acid or an unsaturated fatty acid. So what does that mean? We'll look at that in a sec. So let's look at a couple more important lipids. So here's the lipid type. So a neutral fat, that's what we just looked at. You could have a phospholipid, steroids, would include cholesterol, bile salts, vitamin D, sex hormones, estrogen and progesterone. are derived from cholesterol, and they're lipids. Here's some fat-soluble vitamins. So these vitamins can only dissolve in fat. So if you had a completely fat-free diet, you would not be able to hold on to the fat-soluble vitamins A, D, E, and K, or A, E, and K in this cartoon. All right, so back to saturated and unsaturated fat. So this is a cartoon of a saturated fat. So in a saturated fat, the definition is that their chains, these fatty acid chains here, contain only single nonpolar covalent bonds. So they're straight. So see how so each one of these little points, that's a carbon, and then coming off carbon, you got 2 hydrogen atoms along the whole chain. I'm not going to draw it all in, but if you are a saturated fat, that means your chains are all have single bonds. So each one of these is a single bond. Here's single bond, single bond, single bond. If they are single bonds, then these chains can pack really tight because they're not kinked. They're not like bent like this. If it was bent like this here, then you wouldn't be able to pack this chain close to this chain if the upper chain was bent. So they're not bent. They are not bent. Not bent. So that means you can pack these chains close together. That means if you're packing them close together, saturated fat is going to be solid at room temperature. So butter, as you know, is solid at room temperature. Well, maybe not in Tucson room temperature, but. And then unsaturated fat, check it out. Look at this. There's a double bond here. When you have a double bond in these fatty acid tails, that means they kink or they bend, and now these tails can't sit directly on top of each other and be compact. And so at room temperature, if you are a unsaturated fat molecule, you are not solid at room temperature. Okay, so saturation just means that every single one of these carbons in these fatty acid tails, every single one of these carbons has a single bond. That means that this molecule is saturated. If you have a double bond, you are unsaturated. You've probably heard of trans fats. Those are oils that have been solidified by the addition of hydrogen atoms at the double bond. So they took, if they're trans fats, then you can chemically add some hydrogens here and get rid of that double bond, make this tail go straight out, and then you can get a solid fat. They're not so good for you, I guess. They increase the risk of heart disease. I'm not sure how, molecularly how, but there is a correlation between trans fats and heart disease. You probably heard of these omega-3 fatty acids. Oh, you got to get your omega-3s. So you find them in cold water fish, plant sources. You can get them in flaxseed. Oh, delicious pumpkin seeds, chia seeds, walnuts, anything with soy. Omega-3 fatty acids are delicious, and they decrease the risk of heart disease. And this is how you look at it. So here's an unsaturated fatty acids. So it's unsaturated because it has a double bond, and it's got an omega end. So if you are an omega-3 fatty acid, you would look like this. Phospholipids, these are really important because they are in your cell membranes, in your phospholipid bilayer that we'll talk about when we talk about cells. So a phospholipid, you have this phosphorus containing head group. It's polar because it's got a charge on it, so there's a negative charge right there. And then it's got the glycerol backbone, like our triglyceride did. And then instead of 3 fatty acid chains, it's only got 2 fatty acid chains. And this one looks like it has a double bond, so it's unsaturated and it kinks. So phospholipids in general contain 2 fatty acids rather than 3. We talked about that. We said this phosphorus head group has charge on it. So if it has a charge on it, it's going to react with water. So this negative oxygen is going to react with the positive hydrogens in a water molecule. Fatty acid chains or tails are non-polar, so they don't react with water. So it's a polar head group, but the tail, so it's polar head group, so that means it does react with water. So the polar head group will react with water, but these fatty acid tails are non-polar. That means they will not react with water. So here's our cartoon of our phospholipid. It's got the hydrophilic head, so it likes water, and then it's got these hydrophobic or afraid of water tails, hydrophobic tails. And then here's what it looks like with the atoms in it. So here's the polar head group. Here's the glycerol backbone. Sorry, this whole thing is polar head group. This orange stuff is polar backbone. And then here's the non-polar fatty acid chains or tails that don't like water. And then here's the cell membrane. This is called a phospholipid bilayer. So here's the polar head group. It likes water on the outside of the cell. And then here would be on the inside of the cell with another polar head group that likes water. And then all these tails in here don't like water. So they hide from the water in the extracellular environment out here and the intracellular environment in here. They hide from that water in this phospholipid bilayer. Steroids. Steroids. Cholesterol. Here's a simplified structure of cholesterol. This is what it looks like chemically. You have 1, 2, 3, 4 rings that lock up with each other, and then all these functional groups like a methyl group. Steroids are made from cholesterol. Some cholesterol is ingested from animal products. So when we eat meat, your liver will make cholesterol for you. The cholesterol is the basis for all the steroids in your body. So, for example,